All right, so it's 12 o'clock. Welcome everybody to this year's silent auction. Due to the COVID pandemic, it has to be virtual. This is the first time we're having an all virtual silent auction. But yeah, we have to make do with what we have now. So I'm George, I'm the president for Minnesota IFT. And I'm very happy to welcome you all to the silent auction. And without wasting any much time, I would hand over to the silent auction committee to take over the program. I would come in at the end at some point, but yeah, I would hand over to Steve. So you have the floor now.
All right. Thanks, George. Stephen Kaufman here. It's been a while since I've been involved with Minnesota IFT with COVID, et cetera, and work. But today I'm helping out with the silent auction. We've had this event for a number of years. I can't remember how long it's been. It's been quite a while. It was actually started by Dan Kennedy. And we're trying to do it virtually this year. It's not quite the same, but I've got the latest stats from Lisa Polvoni. She's actually the head of our silent auction committee this year. So right now, donation-wise, when you do log in, you can bid on items. We have 8 items that haven't been bid on yet. So I'd encourage you to get there. Tell your friends and other folks involved to please log in, establish an account, and start bidding. But we can also accept donations as part of that login. So there'll be an option for you to donate now, and all you do is log in, give them your email, your phone number, and how much you want to donate, and then it'll ask you. You can either send a check or put it on a credit card, I think, at that time. So we've got some donations: $100 from Rusty Nelson, good old Rusty, $200 from Grain Millers, $100 from Merlin Development. That's who I work for now, by the way, and $100 from IFF. Our goal is $5,000. Bidding ends, I believe, at 8 p.m. this evening. We've got 1,450, so we've got about $3,500 to go. So let's get bidding. And with that, I'm going to turn it over to, is it Becky, or is it who's leading the RE presentation?
Harold Stanislawski.
Oh, okay. Hey, Harold. So we've got the RE folks today presenting its developments in the wheat industry, tracking wheat digestibility. And I am just going to then give it to Harold and sign off, and I'll come in at the very end to give you an update on the auction. So take it away, Harold. Thank you.
All right. Well, thank you, Steve, and welcome to all our listeners out there on a very interesting topic we're going to have today called Developments in the Wheat Industry and Tackling Wheat Digestibility. Just to introduce myself, I'm Harold Stanislawski. I'm a Project Development Director for the Ag Utilization Research Institute. George, let's go to the next slide. All AURI programs and resources overview is where I'll start. Our tagline is agricultural innovation from idea to reality. Next slide. We will have a moderated panel question and answer session at the end. So all the listeners out there, please post your questions in the chat box, and we will get those to the end of our presentation and answer your questions from the experts who are here today. Next slide. A little bit about AURI programming and who we are. Again, AURI stands for the Ag Utilization Research Institute. It's a nonprofit corporation that was created by the Minnesota legislature to increase utilization of Minnesota's rich agricultural resources. And we work in 4 areas: of food, co-products, bio-based products, and renewable energy. And today, of course, we are focusing on food. And 3 things primarily happen within our service. One is commercialization services, which includes private one-to-one projects like you'll hear today, business feasibility, entrepreneur and residency programs. And then we have public initiatives, which are free to the world, and they're applied R&D for the industry, includes ag innovation partnerships to catalyze some of this activity. And then an example today was our AURI Connects projects, where we have industry convenings, public dissemination of research projects, reports, and such. And then our next one will be, we call it Webinar Wednesday, Fields of Innovation. Next slide, George. One of our guiding principles, of course, is to transfer Minnesota's rich agricultural products into sustainable businesses. And we do this by providing affordable access to consumer science, technical expertise, and infrastructure. And we work with a lot of good folks, including the University of Minnesota, Grow North, Good Acre, and all of the ones that you see down there, Department of Ag, and many, many others. It's really a great thing. Next slide. We just want to introduce some of our key people that we have working in food with our services and facilities. And we do have staff expertise in food science, processing, packaging, and food business development. And Jason Robinson is our business development person in the food space and located in the Twin Cities, comes with a rich background in food with experience in General Mills and others. Lolly Okino, our food scientist, and Ben Swanson, our food scientist, to work in the food space, answer lots of questions. And Ashley Harguth is our program manager in food. And everything happens here from hands-on product development guidance, nutrition facts, labeling, regulatory guidance, thermal processing and safety reviews, cold packing, commercial kitchen networking, school meal crediting. And we also have a lab in Marshall, Minnesota that does food meat product lab, food product evaluation and sensory lab, which is brand new to us, and a wet chemistry analytical lab, and that's all located in Marshall. Next slide. So I want you to meet the panel today. We're going to have three really great speakers today. First is Dr. James Anderson, a professor of wheat breeding and genetics, and he'll talk about what's happening with the University of Minnesota with the project at hand. George Honor, Dr. Honor is assistant professor in the Department of Food Science and Nutrition. We're really glad to have him on this project, provided a wonderful set of expertise for us. And then we have from the private sector, Brian LaPlante, Chief Executive Officer of Backwind Foods Incorporated, located out of Fisher, Minnesota, which is right in the heart of the Red River Valley. And Brian, we're really glad to have you. So all the full bios are available in the chat box as well. Next slide. Jim, take it away and let's talk about tackling wheat digestibility, FODMAP and ATI levels and wheat lines.
All right, thank you, Harold. So I'm going to talk about the first part of this project that is done really by George Honor and myself, and also Praveen Bajgeen, who is a postdoc in my lab. So next slide, please, George. So we know that per capita wheat consumption in the US has been going down for a number of years. Next slide. So, and that decline has been a lot of these fad diets. Some have kind of come and gone, but some of these have been around for a while. And what it results in is people removing starches and oftentimes wheat from their diet. And a lot of this is to specifically try to avoid gluten. Next slide. So why are people trying to avoid gluten or wheat altogether? So I think most of this audience knows that gluten is a protein found in the grain of wheat, rye, barley, composed of two major proteins. Celiac disease affects about 1% of Americans. So that's an immune disease. They really can't tolerate any gluten whatsoever because it's going to do damage in the small intestine. And then another very small portion of the population has wheat allergies. But neither celiac or wheat allergies per se can account for this drop in wheat consumption or for the proportion of people in this country and around the world that are trying to go gluten-free. So we're focusing on non-allergy, non-celiac wheat sensitivity, which has the acronym NCWS. So these folks have some sensitivity issues they have this discomfort, gastrointestinal issues when they consume too much wheat, but they don't have celiac or don't have an allergy associated with it. So we can go to the next slide. So if it's not gluten per se, what are possible causes of the non-allergy, non-celiac wheat sensitivity? This project is focusing on two of those components. The first is FODMAPs, which is an acronym for Fermentable, Oligo-Di-Monosaccharides, and Polyols. And you can see all of the components there. So these are, a lot of those easily digested starches. And then there's ATI, or the Amylase Trypsin Inhibitors. Now, other researchers have looked at these components somewhat in wheat, but we wanted to take a closer look in materials in our program and some historic wheats and see if they could be part of the solution for us. Next slide. So first on the FODMAPs, it's not just wheat products that have FODMAPs. And so in the upper right-hand corner, you can see that rice is very low in FODMAPs, and all of these are in grams per serving. The wheat products are on the high end here. Corn is also low. The wheat products, for the most part, are going to be more than 0.5 grams per serving. And what's been shown in the literature is that FODMAPs are best tolerated if they're at less than 0.3 grams per serving. So that's kind of where we're going to try to target our research. And you can see in the lower left-hand panel that there are a lot of other foods, including a lot of fruits and vegetables, that are also high in FODMAPs. So now there's low FODMAP diets that are probably going to have reduced amount of wheat, but also reduced amount of onion or grapefruit, some of these other fruits and vegetables that are also high in FODMAPs. Next slide. So this is a project that's funded by the Minnesota Department of Agriculture. Our partners in this with the University of Minnesota are ARI, Minnesota Wheat Research and Promotion Council, and Backwin Foods, represented by Brian LaPlante. So our long-term goals: try to reduce the discomforts that people have resulting from the consumption of wheat-based products. That will generally improve the health of consumers, and ultimately it's going to make for a stronger wheat industry and increase the profitability for wheat farmers as well. So specific objectives, I'm going to talk just about objective 1, and that is to characterize the variation that we see in FODMAPs and ATIs, and also try to identify genetic markers for them in a panel of ancient heritage and modern wheat varieties from 2 Minnesota locations. The second objective is to explore the use of fermentation as a technique to reduce FODMAPs and ATIs. Brian LaPlante is going to talk about that. And then the third objective is to get this out into industry. How can we implement our findings in the real world, essentially? So for objective 1, we had a panel of 220 ancient heritage and modern wheat varieties grown at 2 locations. This is a very large number, but this is the sort of numbers that geneticists like to work with to really get at those underlying genetic causes. These are not the numbers that a food scientist like George Honor likes to work with. So kudos to George and his lab, his personnel, for handling this large number of samples. Then we did some genetic fingerprinting, got DNA marker data from all of these using a technique called genotyping by sequencing. And then the whole grains were analyzed for their percent FODMAPs and ATIs in George's lab in food science. And finally, we did association mapping to try to get at some of the genetics of these traits. So here's the composition of this panel. So we had what we called heritage wheats. These were those that were released before 1970. So there's, you know, Fife, Thatcher, Canis, you know, a lot of the founder lines of our breeding program are actually present in that group. Modern wheats, and those are released after 1970. That's when the semi-dwarf trait first came in. This is the largest segment because we really wanted to get a good snapshot of what our current breeding material looks like in terms of these traits. And then these other ones were more sort of included as outgroups or out of curiosity to see where they fall. So Durham wheat, we had 5 of them. Some ancient wheats or einkorn, that has just the A genome. Common wheat has the A, B, and genome. So, you know, it's called genetically, it's called a polyploid. It's genetically more complex. So we had 10 of these einkorns, 11 emmers. Emmers are kind of the wild progenitor of Durham wheats. And then synthetic hexaploids. And these are actually made by crossing a Durham wheat that has the A and B genomes with the wild D genome relative of wheat, combine them together, and you have the A, B, and D genome. So this is a process that was done in nature, you know, going back 10,000 years, but these can also be made synthetically nowadays, and that's becoming a more common approach in the world of wheat breeding. So we included a few of those as well. So these 2 panels on the right, these were grown in Crookston and St. Paul. A lot of variation, as you could imagine, some, you know, really extreme heights and heading date, yield. So very, very diverse panel, just phenotypically. So we'll get into some results here. So first of all, we looked at the genetic diversity. So we did genetic fingerprinting, doing some DNA sequencing on them. What we end up with is about 4,000 DNA markers, and we can compare those allelic states for all of those markers for everything in the panel. So this is a 2-dimensional scatter plot. It's called principal components analysis. We're on the x-axis. They get stretched out based on most of the variation, and then they get stretched out on the y-axis with additional variation that's shown. So the farther apart any 2 varieties are on this depiction, the more distantly related they are. So you could broadly group the bread wheats over on the left-hand side, the Durham and synthetic hexaploids near the middle, and the emmers. The einkorns didn't make this graph because this type of analysis really wasn't suitable for them because they're genetically much simpler with only the A genome. So now if we take just the bread wheat panel of the common wheat, so excluding einkorns, Durhams, emmers, synthetic hexaploids, now we can better separate these common wheat lines. And I know you can't read any of these names, but each of those, you know, dots represents the name of a variety. And I could roughly classify that left-hand quadrant as mostly the modern lines. The heritage founder lines are a quite diverse group, but they are separable from the modern wheats. And then the Chinese, the Mexican, and the South American, those are introductions that were made into our region over the years. Some of them go back, you know, several decades, mostly for disease resistance, and they sort of form their own group. So what this tells me is that we have a fairly genetically diverse group of panel that we're looking at here. So here's you know, more than a year's work of George's lab in one slide. So we have FODMAPs, excess fructose in percent on the x-axis, and the ATI levels in percent on the y-axis. So all I'm showing here is 167 common wheat varieties where we have data on both FODMAPs and ATIs. There's a few where we had missing data for whatever reason, they didn't grow well, So this is all on a small amount of grain using HPLC methodology. What we see in the FODMAPs is there's actually a pretty good range going from about 0.3, 0.4 up until 1.2%. Most of the wheats are in that middle area, as you would expect in an analysis like this. But having those, to lower than 0.5, that gives me hope as a breeder that we maybe have something to work with here that we can make crosses with. With the ATIs, we also see a pretty good range. If we get rid of those really low extremes, functionally it's between about 1.5 and 3.8, so more than a twofold variation in ATIs as well. And we've already identified some in this lower left-hand quadrant that have low FODMAPs and low ATIs that are of interest in our breeding program going forward. So we've already made crosses with them to try to introduce these lower ATI, lower FODMAP traits. I should mention that the ATIs are correlated with protein content as well. So a high protein variety will tend to have higher levels of ATI as well. So right now, George is working on getting the protein contents of these so we can use that to, kind of make a correction or look at that relationship a little bit more closely. So again, to remind you where we're at, we're trying to shoot for this 0.3%. George, if you go back quick. So 0.3% on this graph, we don't have too many wheat varieties that are below that, but we've never selected for this trait either. So, the saying in plant breeding is, if you see variation for it, you can select for it. So I'm hopeful that once we start making appropriate crosses, seeing that variation, we can select for it and hopefully drive these levels down even lower. Okay, I think we can go on to the next one. Okay, so here comes the genetic analysis. So what this figure is showing is we've got about 4,000 DNA markers on each of those members of the panel. Wheat has 21 chromosomes, so that's roughly 200 DNA markers per chromosome. So on the x-axis, imagine all of those DNA markers laid out in the order that they are on the chromosomes. And on the y-axis is the strength of association with the FODMAP data and that particular DNA marker on this entire panel. So most of this is just noise, because we know that most of those DNA markers are not going to be associated with a gene affecting FODMAPs. So that's the things that are coming in below 1 on the y-axis. Once things start to get above 2, then it's like, okay, there might be something there. There might be a genetic cause and effect here. And once you get above 3, that's our statistical threshold. Now we say, okay, George has got his cursor on chromosome 14, that blue dot there. That's something that we're going to follow up on and see how much of the variation in FODMAPs that explains. Overall, what this result tells me, though, is we don't have anything really high. I'd get really excited if this y-axis went up to like 9 or 10, and we had 1 or 2 markers, and all of these other ones were 1.5 or less. That's a nice low noise experiment with a really high signal. That would tell me that there's maybe a single gene responsible. So what this is showing is it's probably genetically complex. There's a lot of genes involved, none with a large effect. So it means it's going to take a little bit more time. Doesn't mean we can't do it, though. We can still make progress selecting for these traits. And the next slide shows the same analysis for ATIs, but a little bit better news here, I think. Look at where the 3 is here. We have many more dots above. So each one of those dots is potentially a gene somewhere in the genome that's affecting ATI content. Now, what I believe is going to be the case once we get the protein data is probably half or more of these will just be protein per se. But hopefully we can tease apart some that are actually due to ATIs and not the actual protein value. So hopefully we can make some progress with that also. So to summarize, objective one, we did see a genetically diverse set of wheat lines. We could make those separations based on modern wheats, historic wheats. We did see quite a wide variation in FODMAPs and ATI content. This is only on 1 year of data, and this is based on 2019 crop. We also grew about half the panel in 2020. Those are still being analyzed in George's lab. But the FODMAPs, we see about effectively a threefold variation in the common wheats from 0.4 to 1.2%, and a little bit more than a twofold variation in the ATI. So we've got something to work with. Now, I didn't mention, or I didn't show the einkorns or the emmers. The einkorns were really low in ATIs, 1.3%, so well below the common wheats, and also low in FODMAPs at 0.3%. Emmers were also low in FODMAP. So that means that consuming these products per se, so if somebody is selling a bread or other product made from ancient wheat, there's a pretty good chance it's going to be low FODMAP already. Now, whether we can take advantage of this in our breeding program remains to be seen, because these are at a different ploidy level. So we could make crosses with einkorn, but once we get it back into a common wheat, I'm anticipating that those ATI and FODMAP levels are going to be elevated again. So one thing I was really interested in was this third bullet here, looking at the year of release and seeing if there was a trend towards higher FODMAPs or higher ATIs, just from inadvertent selection that we've been doing in the breeding program, over 130 years of breeding. As of now, I don't see any real red flags. We can find modern wheats that are on the low end of that FODMAP and ATI. We can find modern wheats that are on the high end, and a similar situation for the heritage wheats. So once we get that second year of data from 2020, we can show that data a little more clearly and make a stronger statement. And finally, no genomic region is responsible for a large proportion of the variation for these traits. ATI is probably in a little better situation because we saw a lot of hits up high. So probably a few major genes there. Nevertheless, we should be able to do selection in our breeding program to help drive both of these down. So now I will turn it over to Brian for the fermentation study.
Thank you, Jim. I appreciate that. My name again is Brian LaPlante. I'm with Backwind Foods, and we've been involved in these research projects for a couple years here to start looking at what bakers like myself can do to make wheat cereal grain products friendly for consumers. So one of the things that we wanted to look at within this research project is to look at the type of fermentation model that we use to reduce FODMAPs and ATI activity in the wheat products that are made. And so just to really quickly go over sourdough fermentation that I use, it's a type 1 sourdough fermentation process, which is, your traditional type of process that's been used for thousands of years, which means essentially that a stable symbiotic culture of bacteria and yeasts are perpetually fed and kept going for daily process to inoculate your flour and water for leavening and fermentation. So we use this type 1 sourdough process to prepare the different wheat varieties that were selected in this research project. And we looked at the function of time and what the time would do to the levels of FODMAP and ATI, holding all other variables constant. So we did cover selections that Jim and George gave us for FODMAPs and ATIs. So what we were looking at is low, medium, and high quantities of FODMAPs, and again, low, high, medium for ATIs. And we selected 10 varieties for each of those groups that we're going to analyze. Next slide, George. So this is a picture, of course, from Rolf Hagberg, who does beautiful work. This, in a nutshell, is what my process looks like. In the back is a fermentation jar of culture that is refreshed, and generally about 2 hours after fresh distilled water and flour is added to it, it's biologically very active for fermentation. Bottom right screen is, of course, the whole berry wheat and the flour. And this is a typical French bread that comes from our process. Next slide. So just as a kind of an overview of what we did, again, we looked at the potential for type 1 sourdough to degrade or hopefully even eliminate FODMAPs and ATIs. And we selected 10 varieties from the low, medium, and high FODMAP groups and 10 varieties in the low, medium, and high ATIs. And we did that for both the St. Paul and Crookston locations, and applied the type 1 sourdough fermentation on a 4-hour cycle and a 12-hour cycle. And in commercial type 1 sourdough processing, these are going to be really common fermentation cycles. Some people go much longer cycles, but generally the rule of thumb in bulk fermentation to double your dough size and then get it into bread proof again to double and bake is going to get you anywhere from a 6 to 12-hour cycle, or maybe up to an 18-hour cycle. So we targeted 4 hours and 12 hours to see what the effects are going to be. So you utilize a portion of the type 1 sourdough to be sequestered as a control, so that we have a baseline to work with. And in all of these different fermentation doughs that we did, we also have a control each for each of the 10s, so that prior to fermentation, we have a baseline that establishes the amount of ATI FODMAP in the culture that's used to inoculate the dough, and then the flour from each of the selected varieties. Next slide. So this is a really simplistic view of how a type 1 sourdough process works. Of course, on the left is just a milled flour. This is a base culture flour that we use. That's a North Dakota mill bread flour that's used as a culture maintainer, and is used to create what you see in the middle is an active culture, which when we refresh or backslop our type 1 portion is always kept back. We feed with fresh flour, distilled water, and it becomes extremely biologically active within 2 hours with very highly repeatable results. And on the far right side again, of course, you're going to see a simple dough ball that would have been used as a representative sample for this research project. Next slide. So there's a little version of a dough ball that would be just slightly larger than what is used for George's analysis down in the cities. But when you look at 2 locations, replicating the research, and then keeping a control for each sample, you get quickly up to 600 individual samples that George has to process, that we ended up in our process to fermenting 4 and 12 hours. So it was a lengthy process, but so George is through the process, I believe, right now of freeze drying them and getting them ready for analysis next month. Next slide. I'll cover more of what happens or what we discussed in the fermentation process later in the discussion, and anyone can ask me any questions regarding that if they'd like to follow up on that. Thank you.
So I think we'll go back to Harold.
Okay, thank you, Brian, for that wonderful presentation, and also to Dr. Jim Anderson. I want to get into some questions that have come in, and I want to encourage anyone of the listeners out there who wants to put additional questions in the chat box, please do so now, and we will get to those. But for now, I want to get into a question actually for Brian. And Brian, assuming a positive outcome to the research, can you talk about challenges of educating the consumers regarding potential new products developed within the wheat and or sourdough fermentation issue?
Sure, and largely this is a function of clearing out a lot of noise that's in the media space and with product claims that are made. But we really do need a process of rolling out the information that's gathered here and research that's related to what we're doing here, because as Dr. Anderson alluded to early, there's a tremendous amount of, I don't want to say misinformation, but people have the belief or the notion that they're suffering from wheat allergies or maybe prone to celiac type of results from eating gluten products. And largely this ramp up in consumer perception of why cereal grain products reacts for consumers has to be cleared up. And Monash University, through their low FODMAP diet recommendations, is doing a lot of that, and I believe our research has to follow along that path as well, because certainly the percentage of consumers that have left the market for cereal grain products dwarfs the percentage of people that actually suffer from authentic, genuine celiac and wheat sensitivity issues. So it's imperative we get this information out, because without a doubt, whole grain, cereal grain products are and have been for tens of thousands of years extremely important for human diets.
Well, thank you, Brian. And this next question can go out to Jim and or Brian. It comes from Canada, a participant from Canada. She says, great presentations. I'm joining from Canada. Does the current US wheat variety registration process consider the nutritional qualities of new lines? If not, will this be considered in the future?
I can take that first. So the short answer is no. In fact, we don't have a national wheat variety registration system in the US. It's basically up to companies or universities to put into the market what they think will succeed, and yield is the number one driver. Depending on the market class of wheat, the end use quality characteristics are very important. So in hard red spring wheat that we're talking about today, the protein content is very important. We're selecting for stronger gluten, higher loaf volumes, but we've never looked at FODMAPs or ATIs before. We've never, anecdotally, we've gotten some information on taste, but really it's largely treated as a commodity wheat, and nutritionally just kind of assumed to be, quite equal, frankly, apart from the protein content.
Thank you, Jim. Another question related to this is, how does sprouting wheat or gluten-containing grains fit in here?
Dr. Anor, maybe as a cereal technologist, this is right up your alley. I could speak a little bit to it, but I think your depth of knowledge goes farther in that category.
Okay, well, so when you sprout wheat, you definitely are activating enzymes that potentially has the possibility of reducing the levels of FODMAPs. ATI, not so sure about that because they are proteins, but you definitely would have some enzyme that may be able to break these FODMAPs into or convert it into other sugars that probably can be used or basically just used up by the microorganisms. So yes, sprouting has the potential of reducing FODMAPs, and I think it will be one of the things that we probably can look at in the near future as we continue working on this project.
You know, and I can speak additionally to that, because those are good points, Dr. Noor. I know that, you know, in the industry, sprouted flours are coming into bakery product lines, and I think that's going to be an important differentiation potentially for commercial producers versus small craft artisan type of bakers who might use fermentation to degrade FODMAPs. But if you're not using longer cycles for degradation, sprouting may be an advantage for commercial type processors, in my opinion, to further impact the reduction of FODMAPs.
Yeah, the only thing is we really don't, for most of the processes product that we use wheat for, we don't sprout them. I've seen people sprout wheat for bread, but it's not really a mainstream process we use for making bread. But you probably can replace some amount of your regular wheat with sprouted wheat to still keep the characteristics of your bread code. But yeah, it can be done.
Well, thank you, Brian, and thank you, Dr. Noor, for that answer. The next question will go to Dr. Jim Anderson. And can you clarify how the research findings intend to be used in your work and the ultimate benefits on the breeding side?
Yeah, so, you know, we started into this thinking that we would find some variation within our breeding program, and we in fact did. So now that we know that variation for FODMAP and ATI levels are there, we can start selecting for it, making dedicated crosses to try to drive the levels of FODMAPs and ATIs even lower. And then going forward, we've also initiated a program where we're trying to breed for different higher resistant starch, different amylase levels as well. So trying to integrate all of that into one package while still trying to maintain yield, straw strength, disease resistance, all those other things that growers need to have in an adapted variety.
Thank you. This question could go for either Brian or Dr. Noor, but as the research advances on sourdough fermentation and it improves the digestibility of wheat products further, is there the potential for specialty ingredient manufacturers to play a role in creating sourdough ingredients that larger companies can use to incorporate with their production methods?
I think I'll let Brian start this.
Okay, so coming from a traditional sourdough approach to this, I would be looking at it slightly different by utilizing the entire sourdough as an entire dough and an entire product. But commercial manufacturers are, I'm sure, going to be looking at research that's coming out like this that may in some way be able to impart some reductions in ATI and FODMAP going forward. It's going to be a process that's going to, of course, add additional processes for them or for the milling industry or the byproducts industries, because you're going to have to take the initial flour. It's going to have to go through a fermentation process with selected bacteria and yeasts. You're going to have to put it through a fermentation cycle and then put it through some form of drying process that's not going to ultimately degrade the high gluten qualities that you need in order to make a final product. So theoretically, I'm sure you could take a flour, put it through a process like I do, freeze dry it or some other low temperature process, keep it as a flour that can actually be utilized in industry for an entire sourdough product, kind of as an instant sourdough. But I'm guessing that's going to add a significant cost to the commercial manufacturers.
Well,
Brian, go ahead, George.
Yeah, I would say I'm very happy we are looking at this from the breeding standpoint, because in the first place, the ATIs are not gluten proteins. They are not part of the gluten protein. So hopefully we should be able to significantly reduce levels of ATI without changing the rheological properties of the dough. And secondly, hopefully we can also reduce the levels of FODMAPs in the wheat, again, such that we don't affect the rheological properties of the dough. Now, if we do that, I foresee low FODMAP and low ATI being adopted and used by everybody. I don't really see us developing another specialty product or specialty lines for these wheat products, because if you should ask me, I don't think we should really change the wheat significantly, but we are yet to find out. So hopefully it can be used by everybody.
Thank you, George. Brian, we've got another question for you. You got a lot of questions here. Brian, why is this project important to you as a baker using sourdough fermentation, and what are your ultimate goals with the consumers?
So the reason this is important is, and Dr. Noor and Dr. Anderson have spoken to this. Without having an analysis of the flour itself and understanding the ATI FODMAP quantities or qualities within flour, you don't have anywhere to start from. So you're kind of shooting in the dark as even a sourdough fermentation process. If in fact sourdough fermentation can affect FODMAP and ATI, how long do you have to ferment in order to knock out or reduce those what you might call anti-nutrients? To get a baseline to begin with, so that you can, number one, produce a high quality product with the kind of organoleptic characteristics that you want. You're going to want the flavor. You're going to want the dough to perform well. You need good crumb structure, all those types of characteristics. And if you are starting with a wheat of very, very high ATI FODMAP, is it going to take you 15, 20 hours to knock it out? Or can we start with a wheat that we know is going to have low ATI FODMAP, and you can go through a traditional sourdough model and create a high quality product and still knock those anti-nutrients out? So for me, it's a matter of starting from there, knowing that can we work with lines that are low to begin with an ATI FODMAP, then put them through a traditional sourdough method, really degrade those so that you have high digestibility. But as a sourdough baker, the nutritional aspects are really important to me. All the different things that sourdough does to cereal grains is really important, but I also want to know that my consumer is getting a finished product with the lowest reactivity. So we can do bagels and breads and pizza doughs and pasta, all these things through our sourdough fermentation model. That's really the goal here, is to create a product I'm pretty positive when it hits the consumer's table that I can be sure that it's going to be very low in reactivity.
Brian, again, another question here, but you know, I'm fortunate because I've been fortunate to have tasted your bread, and it's really good. It reminds me of the kinds of bread that I've eaten in Europe when I've been there. If this research supports longer fermentation models and a combination of bacterial and fungi culture sourdough, how does this impact the bakery production models? Because isn't it going to take longer to, you know, to get that bread produced?
Right. So, you know, if you look traditionally throughout history, the type 1 sourdough method was used largely up until, you know, the turn of the 1900s. And then finally with the sequestration of particularly one yeast variety for what is utilized in modern production, we've certainly gone from what once dominated the markets to something now that is almost extremely rare to find sourdough production. I think the research is going to be beneficial to the fledgling sourdough industry in the United States and what once existed across the world. And I think you're going to see a model very similar to the craft brew industry and the specialty alcohol markets, where you're going to see more regional production, local production of bread and bakery products, going back to traditional models. It may not dominate the market, but I think it's going to create a market where consumers who want clean labels, they want the healthiest type of product with a ton of flavor. I think that's going to expand the market niche for people like me and others that want to utilize this technology to make really, really great products. But I'm one of those guys that believes in the ethic of creating high food, and I think high quality food. I think others are going to do that. So we might see something very similar to the craft brew industry that actually has some impact on the market size.
Okay, thank you. Got a very interesting question here, and this could be for Dr. Anderson, Dr. Honora, or Brian. But could you use low protein, say less than 11% wheat, and get high quality breads using sourdough method? Could that reduce the emphasis for growing wheat for strong gluten?
I would say 11% is pretty low, but again, it depends on the composition of the protein you have in there. If you are not really affecting the functional proteins in there, which is the gluten, you probably should be able to do that. And that is one of the reasons why we are interested in expressing the ATI percent as expressing how much ATI in this wheat product as a percent of the protein content, so we don't negatively just reduce the protein content of the wheat. But yeah, you probably can try to do that, but I would think it is pretty low.
And I would add to that, if you're looking really at the consumer as far as what percentage of the market has left the market and what percentage of the market still eats cereal grain products, I'm less concerned about gluten than I am in the reduction of FODMAPs and ATIs. And knowing that, ATIs strictly are not gluten, I want high quality gluten in my product line with low ATI. So that's really a target for me, is to create a high quality sourdough product with a low FODMAP ATI. I'm going to go after the market that is still there, and also would be willing to come back knowing that this research is showing that their reasons for not being able to eat cereal grains, or they have discomfort, isn't really due to gluten, it's due to something else.
Well, thank you. Very good answers there in all of this. This next question might be a little bit similar, but it has a little different twist to it. Fermented foods like sourdough bakery goods are a part of a movement toward clean labels, whole foods designed for healthier food production. How does this trend affect the traditional food producer versus the modern large-scale food producer?
So, just in my opinion on this, I think more and more research that's going towards fermentation, and what we would probably intend to do with the research going beyond this really discusses what happens in the microbiome, what happens in the gut, what happens to the bacteria profiles that maintain human health. And more research is moving that way towards the Mediterranean diets, towards whole grains, towards fermented products, toward clean label. And it's all targeted at the idea of, you keep good bacteria healthy, your health is generally going to be well. And that's just my opinion. I think this trend is going to continue, and products aimed toward the microbiome are going to continue to grow.
Yeah.
Well, thank you, Brian. George, go ahead, please.
Oh, yeah, I was just going to add that you also have to think about the advantages of fermentation. You're basically activating enzymes that could be beneficial to us. You are also basically reducing anti-nutritive factors. And reducing anti-nutritive factors in food is basically increasing the bioavailability of nutrients or minerals in the diet. So fermented foods, I think it is going to go on, as Brian said. People will just have to get used to the taste of fermented foods. That is, I think, one of the barriers for a lot of people. It's acidic, it's sour, it tastes different. But yeah, in countries where they eat a lot of fermented foods, there is research to show they have much more improved health. Yeah.
Harold, can I comment on the previous question on the low protein?
Yes, please.
So the question on, can a low protein 11% do the job? As a commodity, wheat is marketed, and spring wheat in particular, partially based on its protein. So most wheat that's less than 14% is going to get a discount most years. Less than 12% is almost surely going to get a huge discount. We know there's a very strong negative correlation between grain yield and protein content. So I really wish this could be true, that we could, have a wheat with great rheological properties that would make a great loaf of bread at this low protein. But frankly, it's not going to compete very well with those 14% protein wheats. And protein is every grain elevator in the country that accepts wheat, or at least a higher protein type of wheat, hard red spring, hard red winter, is going to have an instrument that can determine that protein within a matter of a minute or two. So that facilitates that marketing on the protein content. Now, going to more of a non-commodity market, as Brian was alluding to, yes, that's possible. And I think you can find things that will make a good loaf of bread in that 11 or 12% range, but not all of them. It's going to be a certain type, and it's going to need some tender, loving care to make a good product.
Well, Dr. Anderson, just following up on that, regarding the wheat breeding, where have we been? What direction are we headed? And what progress has been made on the whole wheat sector?
Yeah, well, that's a big question. So, I like to remind people that, just like the background that you see behind me, the wheats that we're breeding today, I can trace the parentage back, 100 years or more. So that field you're seeing in the background, that's two gentlemen crossing wheat, is from 1895. I could probably guess at the two wheat varieties that they're crossing, and I can find those in the pedigrees of today. What we focused on, over decades of breeding is the yield, the disease resistance, protein content to some extent. It's more or less trying to keep it around that 14%, but have good rheological qualities as well. Some of those older wheats, like Thatcher, was released in 1936, made a great loaf of bread. That's kind of the basis of a lot of the Canadian programs today, because they like that quality so much. And we can find some of those same qualities in our wheats today. So what got me interested in this project is this decrease in wheat consumption. Have we inadvertently done something with FODMAPs, ATIs, the gluten proteins? So this is just kind of a start of it. If we could make low FODMAPs, low ATI wheats mainstream, that's great. If not, if we can support the smaller bakeries with some specialty products, that might be another direction we can go in the future. But definitely more focus on the nutritional aspects, not just the functionality, but nutrition, and probably start looking at taste, amylose content, some other characteristics as well.
Well, thank you, Dr. Anderson. And to sum up our last question before we move into the closing, and I think this is really good because there's an old saying that the consumer is always right. And so why should we as scientists or food entrepreneurs trying to meet consumer demand or consumers care about this research?
Well, I think we said it, Jim said it in one of his slides. If consumers are not happy, it results in a decline in a food product because they are not consuming it. And then it ultimately results in farmers not making money. Now, this has been the long-term goals of this project. So we really have to listen to what consumers say. If you take an example of celiac disease, it's just a small percentage of people who have celiac disease, but it's been blown out into a big deal, said that people who don't even have celiac disease are avoiding it. We don't want wheat to be demonized anymore, as far as I'm concerned. So it is better that we start looking at this now before consumers even start talking about it like they've done for celiac disease, to result in even much more decline in the consumption of wheat. So maybe Jim can add something, and Brian too.
Any final comments from Brian or Dr. Anderson?
No, nothing that I haven't already spoken to, I don't think.
Well, I could just add to that, you know, anecdotally speaking, you know, within my family, within so many folks that I know that suffer different maladies from digestion of different foods, it inevitably goes back to their family doctor, who almost inevitably says, well, one of the first things you might want to consider is cutting out wheat. And we'll start with that. And they go through this process of elimination to see what's causing them a problem. And if they start feeling better from excluding food items, that gets to be the natural responses. I guess I have a problem with wheat. And I think ultimately what that forgets is that whatever we put in our mouth, we're feeding bacteria in our stomach or in our intestine guts. And by not understanding what's happening there, you can be eliminating food group after food group, feeling better for a little while, and then feeling better, and feeling worse later on without realizing that you're really not addressing the problem. And so it's important that I think we understand what it is in certain foods that might be reactive and might be reactive to our microbiome, and figure out what it was typically or traditionally that kept this problem in check, that kept our microbiome healthy. And that's really where I believe this is headed.
Okay, well, thank you, Brian, and thank you, Dr. Anderson and Dr. Honora. We'll go into the closing now if you want to move the slide. I think from what we've heard today from the researchers and from Brian in the private sector is that, you know, the future is bright. I believe that people want to eat bread, that they want to feel good after they've eaten the bread, and they want to include it in their diets if it doesn't disrupt their digestive system. And the research results will benefit the value chain where the wheat industry and the consumers. So as we look at this and we look where the future might be, we know in Australia, they already have FODMAP certification for food. We might say low FODMAP certification for food, that people can know then that, you know, we're under that threshold that Dr. Anderson talked about. And this can create commercial opportunities for entrepreneurs who are in this space, and also change the trend of wheat consumption decline and have people enjoy bread again, become part of their diets, and not have digestive issues. So from that standpoint, we think the future is bright. Let's go to the next slide. So as we look at these commercial opportunities, there are already examples out in the world where this low FODMAP certification is having an effect already. Here in 2018, the LoFo Pantry has a US operation, Mandrilla Group out of the US. They're marketing low FODMAP flour right now. And this is changing the lives of certain consumers who are interested in eating bread again, and maybe using Brian's sourdough fermentation method, which, by the way, I think, Brian, you said that to get the proper fermentation is probably around 6 hours or so. Is that correct?
Yeah, generally, you're going to get a lot achieved in that 6 to 8 hour time frame. Yep. Okay.
Next slide, please. So here's just some examples so we can see that the commercialization is already occurring around the world with different foods that have been introduced that are deemed low FODMAP by Monash University, and then of course by the companies that make those. So these are providing some level of confidence by the consumers. If they consume these, they're going to be palatable with the digestive system that they have. Next slide. So we are considering now with this project, and we are going to put a phase 2 research proposal forward this fall that will continue the work of phase 1. And in phase 1, we selected wheat varieties with low FODMAP and ATI and bred for high amylose and resistant starch. We will investigate the expected glycemic index and characteristics of pasta and bread that will be made from low ATI and FODMAP wheat with high amylose and resistant starch. and then determine how the consumption of these pastas and bread made from these areas will ultimately affect the gut microbiome. So we're going to attempt to establish a pathway for industry to implement the research outcomes as well. And I want to bring to everyone's attention that the USDA has a program called Process Verified. So if you are a baker and you're interested in using, say, Brian's sourdough fermentation process, and you can then prove that you have reduced the components that are necessary, you could potentially verify that in a process and get a USDA label on your product for the value chain and commercialization efforts of your product. That could be valuable to a lot of food buyers into certain markets. And I think that's something that will be certainly looked at very strongly in phase 2 and so forth. And then, of course, we have the low FODMAP certification in Australia as an example to go by. Next slide. And this is an example of Monash University's low FODMAP certified symbol. But what was interesting here, folks, is that we already have certain breads that are listed here, from deli-style seeded breads to ciabatta rolls to hot dog rolls and others that have already been certified low FODMAP by the Monash folks. And that's a lot of bread products there. Next slide. So a call to action, in your opinion, and again, I'll put this out toward the panel, what would be needed to successfully market a product taking advantage of this research? You can respond in the chat box as a listener, but we also can, Brian, perhaps you want to make one comment here.
Yeah, so I mean, this is exactly what I'll be doing as we roll into business is taking this research in our sourdough processes to get bagels and bread and cookies, those types of products into the marketplace. And it's largely going to be communicating what sourdough fermentation does, what we can do with a selection of wheat and wheat products to make products that are low in FODMAP and ATI. But largely it's a communication to our specialized markets that this is why this type of bread, this type of bakery product is important to you as a consumer. But it is an education process as well as creating a product that is very much sought after in the market for its quality and flavor benefits too. So it's going to be a lot of specialized marketing and direct communication with the market, and let word of mouth do its magic.
Thank you, Brian. Let's go to the next slide, George. Just a little bit on resources and upcoming events here. To learn more and follow this research, you can find it at auri.org/agri. All the information is there. And you can also join AURI and the University of Minnesota for release of final research findings at the AURI Fields of Innovation webinar that will be happening June 25th, again at 12 o'clock. So stay tuned for registration details on that. Next slide. So we want to thank you for joining us. Next slide, George. And I think any questions that you got, you certainly can relate them to me. I'll be happy to pass them on to the researchers to answer those questions. But I do want to again thank the Minnesota Department of Agriculture for the funding for this work and research and project, and the Minnesota Wheat Research and Promotion Council that provides checkoff dollars for research as well and has been a valuable partner in this process. And of course, the powerful University of Minnesota and all the wheat breeding that they do. My team at AURI, and of course, we can't get anywhere if we don't have the private sector implementing some of this stuff and actually driving it. So thank you, Brian, with Backwind Foods for all the good work you've done. So with that, I want to turn it over to back to Steve Kaufman for the final word.
All right. I'm getting going here. You'll see me pop up in a minute.
Yep.
Here I am. Okay. So latest stats on the auction, we're a little light, guys. We need about $3,500 more dollars either through bidding up items or donating. So if you can, go to your management, tell them what a great thing it is to fund food science education, where all our scientists, researchers, nutritionists come from, the people that can put on programs like this. I thought it was a really interesting presentation, touching on a lot of things, science, genetics, and consumer perceptions about things, and health and nutritional factors in our lives. So it's a great presentation. A couple things that are on the auction. The Givaudan Golf package has 0 bids. I already went for the, sorry, I'm losing it, the other golf package. Whoever put it out there, come on, someone help me out. FONA, I bid on the FONA golf package. And then we still have 3 openings for Dan Kennedy's ukulele lessons. So, as you know, Dan Kennedy initiated the silent auction fundraiser for scholarships way back in the day. Had to be in the 90s sometime. I can't even remember when it was exactly, but he started it. He's a great guy. Getting to know Dan through some ukulele lessons would be a great personal development for everyone. So I encourage everyone to do that. So that's about it. It's been kind of a tough year, kind of hard to do this virtually. The auction is going to be going till this evening, 8 p.m. So again, I would encourage organizations to go the donation route if you can. Bother people, or after a little while you feel guilty, go visit the site. And I put the link up on the chat area. So it's just basically minnesotaift.org, and then you'll be basically directed to the auction site. So I just have to type in a email, a username, and I think it is only for this this event, and then it'll go away. So again, thanks to all the volunteers who helped put this together, to Ari for stepping in. I think it was a really interesting panel discussion. Had 25 attendees as far as I can tell. Yeah, we had 25, a couple dropped off. At any rate, it was great. All right, George, back to you. I guess you're doing the wrap up.
Yeah, thank you, Steve. I want to thank everybody again for being of this silent auction event. As Steve said, it's been very challenging, and this is our first online silent auction, virtual silent auction. But yeah, I want to thank all of you. Please go to the Minnesota IFT website. It's minnesotaift.org, and bid on the items. You have very fun stuff there. And also keep in mind that by bidding on these items, you are contributing to students' scholarships. So with that, I would like to bring this meeting to a close. The meeting was recorded, and it will be available on the Minnesota IFT website and also on the website of ARI as well. So thank you very much, and see you all guys later. Bye.